between the pia mater and the arachnoid mater. And remember, we have those little spidery components. In this subarachnoid space, we have the cerebrospinal fluid. And the cerebrospinal fluid gains access to the subarachnoid space via 3 openings. But remember from lab on Monday what those 3 openings would be? Actually, the 4th one got the bottom spinal cord. In the 4th ventricle, we have the 2 lateral apertures and the median aperture. And so the cerebrospinal fluid can continue down to the tip of the spinal cord and exit there. But most of it's going to exit from the 4th ventricle and move into this subarachnoid space. Because that fluid comes from the blood via the capillaries in the pia mater, it has to be returned to the blood to have this continuous cycling. It starts to build up. And so on the surface of the brain, we have veins. And these veins are going to travel in the pia mater and will pass through the arachnoid mater as they pass into our sinuses. So that's what I'm going to draw next. And I'll draw the dura mater as the tough layer, so it's going to be black, and it's adherent to the arachnoid mater. However, in this region, the dura mater is split to form a channel. And down here, it is split to form a channel. The vessels, the veins that are in the pia mater will open up into this space. And the arachnoid mater, or purple, will fold into that space as well, creating an arachnoid villus. So we just take that one area and draw it slightly larger. This would be the superior sagittal sinus. Here would be the arachnoid mater. And here would be some of the veins coming from the surface of the pia mater. So this is known as an arachnoid villus or granulation. These are cerebral veins. So these 2 fluids flow together into the superior sagittal sinus. The arachnoid villus is adding CSF, the superior sagittal sinus, and the cerebral veins are adding, of course, venous blood. That's what we'll see in our next unit, our cardiovascular unit. This material, this fluid, leaves the skull at the jugular foramen and enters into the venous blood at the internal jugular vein. And that's the formation of the internal jugular vein. So let's look at a few images, and then we'll move on to the brain itself. Give you a moment to finish copying those. There are additional sinuses There's the inferior sagittal sinus here, associated with the cerebellum, and the occipital lobe. There are sinuses, but again, we'll pick those up when we do the cardiovascular system. So this first image is a coronal or frontal section through the brain, and you're seeing the skull removed, and similar to the diagram that I drew on the left. So here is the dura mater. And the skull bone, the frontal bone and parietal bone. And the dura mater is split to create this largest channel right here. We'll talk about this space as called a longitudinal fissure between the two cerebral hemispheres. Here would be the lateral ventricle. And here is the pia mater right along the surface of the brain with those spidery web structures of the arachnoid matter. Here are the cerebral veins. So blood, venous blood from the brain is returning to the surface and traveling in the pia mater, as in the CSF in the subarachnoid space. So again, this is just a lateral view of those arachnoid villi appearance here at the top. The dura mater peeled off to show how all the veins are flowing towards the sinuses. Superior sagittal, this is from a natural cadaver. And the superior sagittal sinus from the posterior view. These are the other sinuses that we'll talk about in our cardiovascular system. But this is the diagram that I've drawn larger. So here we have the dura mater split to form the superior sagittal sinus, which is containing both CSF and venous blood. And here you would have the cerebral veins, and these are the arachnoid villi. Okay, they actually can be large enough to create depressions in the skull. The skull is hardening and ossifying, little depressions can be formed in them by the pressure of the fluid in these arachnoid villi. So in that dura mater, here would be the pattern of the superior sagittal sinus that you saw in lab. All right, so let's look at the flow then of cerebral spinal fluid. I believe you have a diagram like this or similar to this in your lecture notes, or like this. And so here is an arrangement of the ependymal cells with the capillaries. So fluid from the capillaries will leave the capillaries, and as it passes through, the red blood cells will stay behind. Some of the white blood cells will come with it, so it's not uncommon to find white blood cells in the CSF, but you should find red blood cells. And as it passes through the ependymal cells, it's modified. So the protein content, the glucose, the sodium content is not the same as in plasma. And it enters into the lateral ventricle and flows. So this diagram illustrates the flow. So it flows from each lateral ventricle through the interventricular foramen. More CSF is made from the choroid plexus in the roof of the third ventricle. That then flows from the third ventricle through the cerebral aqueduct into the fourth ventricle, and either flows all the way down to the tip of the spinal cord and out, or it leads to the lateral and median apertures, and now is in the subarachnoid space. And then from there, exits via the arachnoid villi to the sinuses. This diagram illustrates the same pathway. I don't know if it's clear or not, but you kind of see that sequence. So choose to be able to follow the flow in sequence through those different components. All right, now let's move to the brain. And when we talk about the brain, what we're referring to typically, for the most part, are the cerebral hemispheres. This is an actual human brain. And when I was in high school, our biology class took a trip I went to high school in Maryland, just about a mile from Washington, D.C., and we took a trip to Georgetown University Medical School. And I remember two things from that trip. One was that they were in short supply of cadavers, and they were paying people ahead of time about $1,500 if they would sign their body away to science. And I remember joking with my friends, well, if they really need bodies, then you end up dying early. The other memory that I have was I thought he was an older gentleman. He was probably in his late 30s to early 40s. He was an engineer, and he had gotten bored with engineering and decided to go to medical school. And he was sitting in this room all by himself with a brain, learning the names of all of these gyri. So when I started teaching at the medical school, or I first took the neuroanatomy, and then I finished my doctorate, went back and taught at the medical school for a few years at Davis as a 2 quarters full-time and 1 quarter, 3/4 time. The students were not learning the names of all those gyri. All right, there's so much more that's been added to the cell biology of medicine that they've cut back on some of that stuff. So the cadavers that they dissect in the fall, the brains are removed, and they literally shave them down with tongue boards, those flat popsicle-looking sticks. So after they learn the outside and some of the gyri, but not all of them, they proceeded we had them on like breadboards, and the students would scrape away with these tongue blades the gray matter, looking for the white. So you go through from gray matter to white matter. By the time they were done with that lab, this individual's brain was a pile of shavings on. It was the weirdest transition that I've experienced. So we will learn some of these gyri, all right, and we will learn the fissures and sulci. So first of all, what do we mean by those terms? The sulci, as I indicated when we were talking about the spinal cord, are shallow folds. So each of these grooves that you see right here, running, this looks like brain coral, or brain coral looks like brain. These grooves that you see here are shallow folds. This is how we get additional gray matter, it's how we get additional neurons. So when we were doing the neuroembryology on Monday, the last step was this increased folding that occurs, and is one of the major differences when you look at the anatomy of brains of pigs or cats or mice or whatever, is the degree of folding that is present. So those are shallow folds, and the gyrus is the mountain, if you will, or the hill that is created between two valleys. So the valleys are the sulci, and the gyrus is that hill created in the fold. So this would be a gyrus, this would be a sulcus. So 2 sulci and a gyrus in the center. When we talk about the brain, the gray matter is in 2 locations. It's on the surface of the brain rather than in the center, like we talked about with the H for the spinal cord. So it's on the surface of the brain, and it's also gray matter in islands or nuclei within the brain. Remember, nuclei in the central nervous system are collections of cell bodies, and outside the central nervous system, they're ganglia. So we have cerebral nuclei within the brain as isolated islands of gray matter, and then we have it on the outer cortex as well. And then a fissure are the deeper grooves. So this is a longitudinal fissure right here. So separating the 2 hemispheres is the longitudinal fissure. And then on the lateral aspect, we have these grooves here that separate the occipital lobe from the parietal lobe. And these are identified as sulci. They're a little bit deeper than these sulci, but not as deep as the fissure. So the cerebral hemispheres are the telencephalon of our prosencephalon. And those are the lobes. We're going to go through each lobe and talk about its function. Frontal lobe, parietal lobe, temporal lobe, and so on. Do you remember the component of the prosencephalon? Remember it divided into 2 vesicles? We had the telencephalon, the cerebral hemispheres, that grows so rapidly it covers over the other component of the prosencephalon. Remember what that was called? It's this area of the brain right here.
Diencephalon.
Diencephalon. Okay, and this is what forms the parameters of our third ventricle, the thalamus and hypothalamus. So we're going to focus first on the cerebral hemispheres, and then we'll move to thalamus and hypothalamus. So the cerebral hemispheres are divided into the lobes, the two hemispheres separated by the longitudinal fissure, and then the frontal lobe and parietal lobe are separated by the central sulcus, which is this groove right here. Okay. Approximately, this is kind of tilted forward, but this central sulcus is approximately not quite 2/3, but more than halfway back. On the human brain, it's really easy to find because the central sulcus forms a groove that actually passes over into the medial surface. All right? And on our plastic brains, they don't do that. So in lab, I'll show you where the central sulcus is, and we'll talk about the gyri around it in just a moment. This sulcus is not in your lab manual, but I will add it to it, and it's the parietal occipital sulcus. Obviously, by its name, it separates the parietal lobe from the occipital lobe. That's at the bottom of your lecture notes. And it's difficult to see on this view. You can only see it part way, here and here. So where I will tag it will be on the medial surface of the brain. And then from the lateral view, we have the lateral sulcus, which is barely seen right here. So let me find another image. The lateral sulcus functions to separate, get to it here. This is the lateral sulcus right here. So it's primarily associated with the temporal lobe, separates the temporal lobe from the frontal lobe and a little bit of the parietal lobe. Posteriorly, parietal and temporal lobe are indistinguishable. Okay, there's not a real good sulcus that separates them. So those are the sulci and the fissure, the longitudinal fissure. And now let's look at some other terms. And we talk about the fibers that separate the white matter. And there are three types of white matter, three types of fibers. So I'm going to raise the screen and diagram these on the board. We'll come back and look at them. So on this cross-section, coronal section here, this would be a gyrus. Sorry, this would be the gyrus here. So that would be a gyrus, and then this space right here would be a sulcus. And this space here between the two hemispheres is our longitudinal fissure. The location of gray matter is thrown into folds. So along the gyri and the sulci, we will see the gray matter. And remember why it's gray? Unmyelinated. And what part of our typical neuron is unmyelinated?
Cell body.
Cell body. All right, so this is where we find motor cell bodies and sensory cell bodies and integrating bodies. And then if I draw this around here. If I draw in our ventricles, there would be the lateral ventricle and third ventricle. Then we see additional gray matter associated. If we were in the midsection of the brain, we would see the thalamus, which is gray matter. We would see additional gray matter here associated with movement. And so this is just, these are nuclei, cerebral or basal nuclei. You may have heard of Parkinson's disease. And the globus pallidus, which is what this structure is, damage to that is what is revealed as the tremors. So when we get further into the brain, we'll talk about that, but I just kind of wanted to identify its location. All right, so now looking at the white matter, as I indicated, we have three different types of white matter, depending on their function. And white matter is going to be myelinated nerve fibers. There are some unmyelinated nerve fibers in there, of course. So I'll add that. But the reason it's white is due to the presence of the myelinated components. And the first type that we're talking about are known as association fibers. So we have two types. We have short association fibers, and we have long. Short association fibers are fibers that move from one gyrus to the next. So in the gray matter, we would have the cell body of one neuron, and it would send its axon to the adjacent gyrus. Okay. So that is a short association fiber. So within the same lobe, all right, we see this when we have areas of the frontal lobe. And the frontal lobe is motor and critical thinking, doing math type of a thing. But for motor, it's voluntary motor. And for sequences of movements that you perform over and over, such as walking, or playing a musical instrument, music that you've memorized, you don't have to think, now I have to put my fingers here and move my mouth. Speech is a predetermined movement, but that's in a different area. So let's say for walking, I don't have to decide when I want to flex my flexor muscles and then extend my knee or flex my knee or dorsiflex my foot. That's all pre-programmed. So we have an area of the frontal lobe known as the premotor area that sends neurons to the sends pathways to the neurons that descend my spinal cord to activate cells in my anterior gray horn. That's the primary pathway. But those are activated without my conscious decision when I'm walking from the premotor area. So that takes short association neurons to do that. So from the premotor area to the precentral gyrus is a short association. I'll remind you of that when we look at the frontal lobe. If we look at a longitudinal pattern of the brain, we would have the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. The longitudinal association pattern, let's say that I see somebody's face. I see your face, Amelia, and I know you, and I recognize you, and I know your name. So from the occipital lobe, where my brain is processing the imaging, the name and the recognition and the memory of that is found in the frontal lobe. And so I have long association fibers passing from lobe to lobe, but on the same side of the hemisphere. All right, so short association fibers are gyrus to gyrus within the same lobe and the same hemisphere. Long association fibers are gyrus to gyrus, different lobes, same hemisphere. So from the right occipital lobe to the right frontal lobe, right? They don't cross. So those are association fibers. Commissural fibers are bridges. Remember we had the anterior white commissure in the spinal cord, and we have the gray commissure. So those are bridging from one side to the other. So if we bring back this diagram, actually I can draw it again. So if we look at a coronal section of the brain, in our ventricles. Commissural fibers are in several locations, but the major ones form a band that cross under the longitudinal fissure and cross to the other side. So they're going to be from the same lobe of one hemisphere to the other hemisphere. So frontal lobe to frontal lobe, temporal lobe to temporal lobe, occipital lobe to occipital lobe. So starting in the gray matter, let's say this is the parietal lobe of the left hemisphere. It's going to cross under the longitudinal fissure and go to the opposite side. Okay, so it's going to form a bridge. And this happens across from front to back of the brain so that we have a bridging of fibers that creates a sheet, if you will. So if I were to diagram those commissural fibers on this illustration, say this is the frontal, the frontal lobe. Have that. And so that band of fibers crossing from one side to the other, from frontal lobe to frontal lobe, temporal lobe to temporal lobe, parietal lobe to parietal lobe, occipital lobe to occipital lobe, forms a bridge known as the corpus callosum. So this band of white matter that we see here is formed of commissural fibers. There are smaller anterior commissures and posterior commissures, but this is the major one. And it forms, when we see it in a median section of the brain, it seems to travel this direction, but it's going back and forth like this. So it forms a broad, it actually curves under itself, in the front and the back. And this would be a longitudinal section of the brain. I'll show it to you on the PowerPoints in just a moment. And the fibers are coming out at you. All right, so they would be, these fibers would be formed from frontal lobe to frontal lobe. These fibers would be formed from parietal lobe to parietal lobe. These fibers would be formed from occipital lobe to occipital lobe in that fashion. Okay. And they're slightly folded under each other. Let me show you the third type, and then we'll look at them online on the overhead. The third type are identified as projection fibers. So we have association fibers, we have commissural fibers. Commissural fibers again, same lobe, different hemispheres. Before we leave the corpus callosum, these are the fibers that allow one side of the brain to know what the other side of the brain is doing. So most of you are aware that the right side of the brain controls the voluntary motor on the left side of the body, and vice versa. So there's a really unique experiment, or it's no longer really an experiment, procedure that can be done. Some individuals back in the mid-40s and 50s, to control severe epileptic procedures, had their corpus callosum cut, separating the left and right side of the brains. That procedure is being done again, all right, on a more limited basis, but it's coming back. And so if you take an individual who's had the corpus callosum cut, and you put them in front of a screen, and you show them an image on the right side of the screen, all right, and ask, so it's being seen on the right side of the brain, and ask them with their right hand to reach into a bag and pick up what they see. Remember, that's being controlled by the left side. So what their right hand is feeling is being processed by the left side of the brain, and the control of the right hand is on the left side of the brain. And you ask them to reach into the bag and pick out what they see. they can't do it. They can tell you, I feel an apple, I feel the tennis ball, I feel an orange. But if it's an orange up on the screen, they can't connect that image with what their hand is feeling. Now, if they say, I see an orange, then of course their ear is going to hear that on both sides, and they can pick up the orange. Okay, but that's a real good illustration of how those are separated. All right, the third type of fiber is identified as a projection fiber. And these are ascending and descending fibers. So these fibers are going to go brain to spinal cord or spinal cord to brain. It may not go all the way down to the spinal cord, it just may go to the brain stem. All right? But we talked about sensory fibers as being going towards the central nervous system. So if we have ascending fibers, they're going to stop in the thalamus. And then those are going to go like that. Or if we have a motor fiber, obviously this would be a little bit farther forward because the motor fibers are in the frontal lobe, not the parietal lobe. But we would have those motor fibers descending all the way down to the anterior great horn. They're going to cross over and descend. So information is going superior to inferior. And those are our projection fibers. So let's take a look at this on the overhead. This is a longitudinal view of the brain, and from gyrus to gyrus, we're seeing the short association fibers, all right, within one lobe, from one gyrus to the next. And then here are the longitudinal, the long association fibers. So passing from the temporal lobe to the frontal lobe, we use these for memory formation. Temporal lobe to occipital lobe to frontal lobe are these longitudinal association fibers. There are areas within each of the lobes called association areas that we'll talk about as we talk about the lobes. And if there's damage to these association fibers, you lose some of that association function. So the primary function is there, all right? So if there's a damage, say, in the parietal area merging with the occipital, you can see a person, know it's a person, but not recognize their face. Jane Goodall, the woman who studied gorillas, has this disorder where she sees, she's not blind, all right, but when she sees a face, she doesn't recognize features. So she doesn't walk into buildings, but the meaning is lost of what she's looking at. Okay. Which is really paradoxical considering how much she learned from the gorillas by recognizing individual gorillas and learning their behavior. So here is some of that scraping I told you about with the tongue blade. And so you can see these longitudinal association fibers here and the short association fibers creating these ridges here. A little easier to see in the diagram of that where the shadowing is occurring. Okay, commissural fibers. So notice on the right-hand diagram how we have fibers going, as I indicated a few moments ago, from the frontal lobe to frontal lobe, parietal to parietal, here's occipital to occipital. So these fibers are bridging from one side of the brain to the other. Right. This is another commissure, not the one major one that we look at, the posterior commissure, but this one is called the corpus callosum. When we look at a sagittal view of the brain, this is the corpus callosum right here. So these fibers here be going from in and out of the screen, frontal lobe to frontal lobe. Again, the fibers are not going in this direction, but they have been cut in cross section, going from side to side. So here's a view of those fibers in a diagram. Okay, and then this is just what I was illustrating about what happens when individuals lose that corpus callosum is cut. So when we do the sheep brain, this space right here is already an open space. The pia mater is on this medial surface of the brain. And when we cut the brain in half, the first thing we have to cut is through the corpus callosum. We would then be in the third ventricle, right here. And we don't have to cut that, because remember, that's already a space. All right? But you can actually separate the sheep brain slightly through the longitudinal fissure, look down, and see that corpus callosum. So these are the projection fibers. So they are traveling primarily from the frontal and parietal lobes. From the frontal lobe, it would be down, going up to the parietal lobe for pain, touch, temperature, that type of processing. All right. So now let's look at, I'm going to leave the screen up here. Let's look at the individual lobes and talk about their functions. When we talked about the spinal cord, and we'll see when we get back from spring break with the brainstem, remember I said that the trend is that anterior structures or ventral structures are motor in function, and posterior structures are more likely to be sensory in function. Right? So when we look at the brain, the frontal lobe is certainly our reasoning and logic and personality part of the brain, but it's also the site of voluntary motor. Parietal, temporal, and occipital lobes are all sensory. Okay? So most of the brain is involved in processing sensory components. The frontal lobe is separated from the parietal lobe by the central sulcus, from the temporal lobe by the lateral sulcus. The fold just anterior to the central sulcus is identified as the precentral gyrus. So fold behind it, part of the parietal lobe, is the postcentral gyrus. If you don't know where the central sulcus is, then it's hard to identify the precentral gyrus and postcentral gyrus. In our plastic models in lab, I do not ask you to find the central sulcus. All right, I will find it for you. So obviously, if I have an arrow within the central sulcus anywhere along this lateral aspect of the brain, the only sulcus that you need to identify there is the central sulcus. Now, what if I just tag the gyrus? That's a little bit more problematic, because you have to know where the central sulcus is here or here to know whether this is the precentral gyrus. So I will either use a brain in which the central sulcus has been indicated or outlined, or one of the questions I will ask is to name that sulcus. So if I ask you to name that sulcus and the gyrus, then you can look anterior or posterior to it and identify it as the precentral gyrus or the postcentral gyrus. So when we look at the structures of the frontal lobe, precentral gyrus is the location of our upper motor neurons. Now, when I was diagramming the spinal cord, let me do this over here on the side again. The motor neurons descended down the spinal cord, and where did they end up? From where I want to extend my fingers. I decide that I'm going to extend my fingers. Excuse me for a moment. When I decide I'm going to extend my fingers, that is a voluntary movement. So that thought originates in my frontal lobe. Projection fibers send that information down to my spinal cord. And where is the synapse going to occur? Where do I find voluntary neurons in my spinal cord? What nucleus? What nuclei? What's the gray matter of the spinal cord that is motor? Anterior gray horn. All right. So if this is my brain and spinal cord, ventricular system, I'm going to move the left side of my body. That information, that thought process, is going to activate motor neurons in the precentral gyrus of my frontal lobe. That's what the precentral gyrus is made up of, our upper motor neurons. Okay. Those upper motor neurons send an axon down to the brain. As we'll see after spring break, that's going to cross over in the brainstem. And now it's going to descend in the lateral spinal cord as a motor pathway. When it gets to the level where the muscle is that I want to move, it's going to go and stop in the anterior gray horn, and then cell bodies, which are unmyelinated, form the anterior gray horn, and they will exit in the ventral root, enter the spinal nerve, and if they're going to any other part of my body than my back, they will pass through the ventral ramus and become part of, say, the femoral nerve or the median nerve or whatever muscle it is that I'm going to move. So this is the upper motor neuron, all right? And the lower motor neuron has its cell body in the anterior gray horn. And that's a 2-neuron pathway. So if I'm going to flex my thigh, I just activated that pathway. Precentral gyrus, upper motor neurons, sent an electrical current, crossed over in my brainstem, went down my spinal cord, ended in the anterior gray horn of my lumbar region of my spinal cord, activated lower motor neurons that went into my femoral nerve to those muscles, and I flexed my thigh. It's that simple a pathway, right? 2 neurons. Now, if there is damage to the upper motor neuron pathway, will I be able to move my thigh if I cut that upper motor neuron pathway? No. I could, if I cut this pathway anywhere along here, if I form a lesion, I have a stroke, or damage to the spinal cord, and cut the upper motor neuron pathway, I'm paralyzed for that muscle movement. Okay? Now, if a sensory neuron comes in, say from my knee-jerk reflex, and forms a reflex, I'll still kick out. In fact, I'll kick out even harder because I will have lost the suppressive neuron information that is always descending. So I'll still be able to move, not voluntarily, but in response to a reflex. Right? So that type of paralysis that occurs is known as spastic paralysis. Because movement can still occur, but not voluntarily, right? As a reflex. The sensory neurons coming into the spinal cord only. Now let's say that I have polio, so I kill the cell bodies of my anterior gray horn, or I have a myelinating disease that damages my lower motor neuron. So I have a lesion that damages the lower motor neuron. Now, my upper motor neuron is intact, but there's no way for that information to get to my muscles. So it's also going to result in paralysis. But that is flaccid. Even if I send the sensory information back, the reflex isn't going to work. All right, so that type of paralysis is identified as flaccid paralysis. Right, so here's the premotor area I was talking about that have the short association neurons. And then down here, we have a similar premotor planning area, but specifically for speech. So it's a part of the frontal lobe right along the lateral sulcus. If you're right-handed, it's found on the left side of the brain. If you're left-handed, you actually have speech areas on both sides. So if you're left-handed and you have a stroke on the right side of the brain or the left side of the brain, it's easier to recover from because you have two components for that motor speech area. This is planning speech. All right, your hearing is fine. You can move your lips, you can move your tongue. There's no paralysis to any of those muscles, but the sequence of movement has been altered. So I'm not thinking when I'm talking to you in lecture or in conversation, I'm not thinking I have to move my lips this direction to say ah or b or q. I just do it. And that's part of that premotor component of speech. So someone who has that area damaged, all right, can't control that specifically. They will say a word. They may say the same word over and over and over when they're trying to say a sentence. They may say a different word than they intended, or they may just make a series of nonsense sounds. There's a variety of different components of that. They can hear perfectly fine. So they know the word they want to say, but it's not the sound that comes from their mouth. It's very frustrating because they hear that sound and they know it's not what they intended. So they try over and over and over. And any form of communication, so they can't write it either. All right, if they try to write it, they have difficulty writing the letter, or they write the wrong letter or the wrong word. If they try to sign it, the same type of problem occurs. So any form of communication, it's not just the speech part that's affected, if Broca's area is affected. And then obviously, of course, we have the prefrontal area here, where we have logical thinking, personality. People who have tumors here can change personalities. The bottom part of the frontal lobe is really important in creating memories, long-term memories, converting short-term memories to long-term memories. All right. There is, when we cut the frontal lobe in cross-section, this is the precentral gyrus that has been cut. And so that upper motor neuron, that cell body that I drew there, doctors and scientists have mapped where that upper motor neuron would be located. So if you're going to move your knee, the upper motor neurons that flex and extend your knee are located right here. Your toes, down along the medial surface of the frontal lobe. Notice that these are the size of the body structures, the number of neurons that are found associated with that. So we have a lot with our hand, a lot with our lips and our face for facial expression and speech. And so this was done. People do not have pain receptors within the brain itself. All right, the dura mater, the meninges, the skull, so local anesthesia is used to open the skull up. And often patients are needed to be conscious when the surgery is going on, so they can ask them to do certain movements, or if they stimulate a part of the brain with the electrode, if they're trying to isolate a region that they want to remove, they don't want to remove the wrong area or too much of the right area. And so these conscious patients who moved parts of their body in response to an electrode stimulus were how these areas were mapped. All right, so let's move to the sensory brain now. And we have the parietal lobe, which is shown shaded here. Parietal lobe is the region shown in blue. And just posterior to the central sulcus, we have the primary sensory gyrus. Primary meaning that's the processing that's occurring. So when I touch the table, the primary processing that we're talking about has to do with pain, touch, temperature, vibration, not hearing, not vision, not balance. And so the sensory neurons follow a 3-neuron pathway. And one neuron, of course, is peripheral. And we'll pull this back again next week, or after spring break, when we talk about the thalamus. That's called the first order neuron. It will synapse in the posterior gray horn if we're talking about pain, touch, and temperature. Balance is a different pathway, but right now I'm focusing on pain, touch, and temperature. And so now we're slightly posterior. All right, this is not going to be the frontal lobe, but the parietal lobe. So we're looking at the post-central gyrus. I'm going to bring in a sensory neuron over here, so we're not facing the same side. The first order neuron, it synapses with another neuron, which crosses over to the opposite side of the brain, and that will stop in the thalamus. We've been talking about the thalamus now for a couple of days. The third ventricle. The thalamus is formed of a whole bunch of cell bodies, and it forms a sensory relay system. So almost all senses, except for smell, their neurons will stop in the thalamus, and from there, the third neuron will go either to occipital lobe or temporal lobe. In the case of pain, touch, and temperature, it ends up in the post-central gyrus of the parietal lobe. And neurons in the post-central gyrus of the parietal lobe process this information and determines this is pain, this is light touch, this is vibration, this is pressure. Okay. So the post-central gyrus is formed of these neurons synapsing with the sensory pathway and doing that processing. And now one of the difference between mind versus brain, right, is the concept, and I've talked about this with a couple of you, but when I touch this table, those touch receptors, remember we talked about Meissner's corpuscles and Pacinian lamellated corpuscles, those receptors, if they're activated, trigger that first order neuron. An electrical current passes along the neurons that I've drawn in green, and finally is processed in the post-central gyrus. Similar to the motor pre-central gyrus, we have a sensory map. So sensory information coming from the knee, or from my foot, or from my lips, ends up on those specific locations of the post-central gyrus. Do I feel that in my brain when I touch the table with my fingertips? Do I feel that sensation of touch in my brain? No. I feel it in my fingertips. There is no physical electrical current or change that goes back to my fingers. All of that chemical processing and changes in electrical current is occurring in my brain, and yet I feel it in my fingers. That process is known as projection, and we can't explain it. It's a learned process. The brain learns that when sensory information reaches that area of the brain, it's come from a particular part of the body. All right? That's why someone experiencing chest pain, heart attack, has pain radiating down their left arm. There's nothing going on in the left arm. Maybe they'll get a headache. There's nothing going on in the head. But nerves from the heart enter into the spinal cord from the chest wall at the same level as from the arm. Sensory nerves from the heart itself travel with the vagus nerve that goes into the skull near the trigeminal nerve. And so in about 17% of the cases of heart attacks, a headache also is present. Not because there's something wrong with the head, but because the brain is projecting that pain to where it thinks it's coming from. Okay? The same thing happens with other senses, more uniquely with vision. So when I see you guys, I don't see you in my occipital lobe. That's where the processing is occurring. I don't even see you in the retina, which is where the activation of the receptors are occurring. I see you at different levels. There's a depth that occurs, and the brain projects that. There's a real cool experiment where they have these glasses that, kind of like a projector or a microscope, the images are reversed left to right and upside down. So you put them on, and the world is flipped over. All you guys are upside down. okay? And the ceiling's down here and the floor's up there. If I were to wear those glasses for 2 or 3 days, my brain learns that that's not the feeling down there, and the image is reversed. Even though I'm still wearing the glasses, the world looks normal until I take the glasses off. And even though I'm not wearing any glasses, my brain has to relearn and remove that adaptation that occurred. All right? This kind of projection is experienced by amputees. So their big toe will hurt or itch even though it's no longer there. All right? And as frustrating as that is, from what I've read, the presence of that phantom pain and feelings, if they have that, makes using prosthetic limbs more successful. Okay? And it's due to the irritation of the nerve endings in the stump. Now, also present within the parietal lobe, we have association areas. So this is known as the general sensory association area. Somesthetic is the same as sensory. And so again, associating, when I touch the tabletop, all right, I recognize it as smooth, or I can touch an orange and recognize it as an orange. So it's taking remembered sensory aspects and associating them with past experiences. Damage to this somesthetic area is associated with damage also in the visual area, and a condition is known as cortical neglect. The damage to the association area. You can stand, say, along Capitol Mall, facing the Capitol. Visually, your eyes work. You can look and see the buildings on the right side of the street, see the buildings on the left side of the street. But if you're asked to describe them, you would describe only one side and act as if the other side wasn't even there. Even though your retina is working, your occipital lobe is working, damage to the somesthetic area doesn't allow you to process that. Okay? So individuals have been tested for this by being given a piece of paper that has a bunch of lines along the paper and asked to make a cross out of each of those, or a T, and they'll go along and they'll do just one half of the page. They can see the other half. There's nothing wrong with their eyes or their occipital lobe. And so you say, well, there's some over here that you've missed. Can you go back and put hash marks through these? And they'll go, oh, okay. All right, I'm done. And they're not trying to be contrary, all right? Their brain is just ignoring that information. If you talk to them, it was really fun when I was teaching at the med school. We had 2 neurologists from Benicia that would come, and one would act as the patient, and he was really boring. And the other guy would be the consulting neurologist. And if you stood on one side of him, he was just the patient, you'd be talking to him, and you just ignore him. And if the guy came around to the other side, all of a sudden, oh, how are you? You know, what's going on? And you talk to him. And so it's that damage is to that association area that Oliver Sacks I told you about last week, the neurologist, talks of the title of one of his books that came out about, I don't know, almost 20 years ago. The title of the book is called The Man Who Mistook His Wife for a Hat. And he's the, typical absent-minded professor walking into a telephone pole and saying, I'm sorry, and everything. And his wife took him to see the doctor, and he took off his hat and jacket and coat and shirt, was examined, and the doctor said, okay, put your clothes on and let's go into the office and talk. And so they talked, and the doctor said, okay, well, I'll contact you. So he goes to put his overcoat on, and his hat's hanging on the wall. And instead of going over and getting his hat, he goes to his wife and tries to lift her head off of her body because he's got that missing. He can see it's a head, but he doesn't have the association of what it actually is. Okay, so it's a weird kind of, and it varies from person to person. We also have within the inferior aspect of the parietal lobe the gustatory or taste area and smell on the deep surface of this region. Okay, temporal lobe. Most of us associate the temporal lobe with hearing. And in the primary auditory area, what you see there in the deep red, is hearing. So when I make a sound, the vestibulocochlear nerve goes to the thalamus. From the thalamus, nerve fibers end up in the primary auditory cortex, and that's processed as a sound. All right, the association area around it allows you to recognize that sound as a dog barking, your dog or somebody else's dog, okay, as the note a high C, but not just that, but whether it's a violin playing it or a person singing it. So those are the types of associations that occur with that. In addition, deep within the temporal lobe on its deep surface folds up and coils around. It reminded somebody of a tail of a seahorse. And so that gray matter is called the hippocampus. We'll actually see it. I'll show you how to find it in the sheep's brain. And that is important for memory, for the creation of short-term memory. You've probably had the experience where all you have to hear is the first note, maybe 2 notes of a song, and your brain fills in the rest of it. All right, so that's stored in memory, also in the association area, but not just sound, all right, but when we read things, when we do things, short association fibers, or actually long association fibers, but they're short, long association fibers, go from the hippocampus to the hypothalamus. When we do the brain stem, we'll talk about that part. And reverberating circuits, all right, help to lay down short-term memory. So wait, within 5 minutes. Long association fibers from the hypothalamus to the bottom of the frontal lobe convert that to long-term memory. But you have to have short-term memory to create the long-term memory. So I just want you to recognize the hippocampus as being a structure within the temporal lobe important for memory. Okay? One of the early signs of this was a sailor who liked to build models. And this was shortly after World War II. And super glue wasn't available. And so in creating the plane models and ship models, he had to hold the structures together for a few minutes before he set them down. And his roommate loved to tease him. Of course, he couldn't do anything while he was holding the models. And so he'd poke them and he'd jab them, and the guy couldn't do anything. So one day the guy got really frustrated, and he whirled around just as the guy was going to poke him with a sharp, something like an ice pick. And it went right through the thin part of his temporal bone and into his hippocampus. And even though it was just on one side, we do have sightedness to brain, and he lost his short-term memory. So the typical, you know, hello, how are you, here's my name, and then 5 minutes later acts like he's never seen you before. Remembered the president when he was, you know, first joined the Navy and all of his childhood memories, but didn't have the short-term memory. It's in the hippocampus and Alzheimer's. We first start to see some of the damages from Alzheimer's in the laying down of that long-term memory. Yes?
So if you have damage to the hippocampus and you can't form short-term memories, from the onset of that damage, will you never form long-term memories afterwards?
Yes, in all likelihood. Okay, with Alzheimer's, of course, you actually start to lose long-term memories too. But it's the short-term memories. It's where did, you know, wandering outside, not being able to, what did I do last? You know, saying that it's not the same as dementia, and it's, you know, your dad starts telling you the same stories over and over and over. You know, I've heard that before, kind of a thing. But it's the, you know, eating, not remembering that you ate breakfast, kind of a thing. You know, that type of short-term memory, and it progresses. All right, occipital lobe. It's also sensory, and it's a visual sensory area. So we have a primary sensory area where we process the electrical currents coming from your retina. The neurons go to the thalamus. From the thalamus, they go to the occipital lobe and are processed as images. And then we have the association areas where those images are associated with previous images or with the frontal lobe or with the temporal lobe to create memories. We can trigger just the neurons in the occipital area and create light images on an individual's retina. So this was shown experimentally in several individuals who were blind. Optic nerve didn't work, the occipital lobe did, and electrodes were implanted in the occipital lobe, connected to a computer, that would scan images from a page. And instead of, there's computers that will read, they'll take a book and read it in a computer voice. But instead of reading, these images converted the words into braille, into pinpoint, into electrical currents, creating pinpoints of light within that individual's brain. Now you'd think, okay, why not just read it out loud and be done with it? But in the experiment, they were seeing the patterns of braille lights. Instead of touching them with their fingers, they were actually seeing them created in the occipital lobe, not through the optic nerve, by the activation of specific regions of the gray matter of the occipital lobe through electrical activation. Just like electrical activation of the precentral gyrus would cause muscles to contract, or electrical activation of the postcentral gyrus would cause a person to say, I feel this in my finger, or I feel this in my toe, or the back of my knee is tingly. And so specific regions of the visual cortex, primary visual cortex, are associated with specific creations of images or light processing. All right, there is between the lateral, we pull down the lateral sulcus. The brain grows so rapidly that the temporal lobe and the parietal lobe grow over and cover up part of the gray area of the cortex. So this is a cortical area. It's covered with pia matter. It's not deep within the brain. It's not nuclei. It's the surface of the brain. It's thrown into gyri and folds, but it is covered up by the growth of the parietal lobe and the occipital lobe. So if we were to look at it from a cross-section, or not a cross-section, but a frontal section through the brain. So here's a longitudinal fissure. We have our gyri. Here's the lateral sulcus between parietal lobe and frontal lobe. So lateral sulcus, parietal lobe, temporal lobe. This structure here would be the intellect. Okay? So if you could open those up and look on the surface, this would be covered with pia matter and is a cortical area of the brain. It's a really weird area. It's an area involved with making judgment calls. So whether you like semi-sweet chocolate or dark chocolate, okay, going to clean out the refrigerator and smelling a carrot or seeing a carrot and deciding whether it's rotten, whether to throw it out or not. Seeing someone cheating on an exam and deciding whether or not to tell on them. So it's a variety of judgment calls that occur in the intellect. Okay? I'll show you, we have brain models where you can pull the brain stem out of the brain, and on the top part of that is the intellect. Okay, so I'll show you where that is in lab, and Dr. Lancaster can show you that. So here on our human brain, the wedge of frontal lobe and temporal lobe have been removed, and you can see there's still cortical folding, okay, on the surface of the intellect. All right. So before we leave, we talked about the motor area for speech. Within the temporal and parietal lobe, kind of overlapping both temporal and parietal lobe, we have the sensory area for speech. And this is known as the general speech area or Wernicke's area. And so this is kind of the other side of the coin. It's probably listed under temporal or parietal lobe in your lecture notes. And it's probably, I don't know which one would be more frustrating. Wernicke's, and there are individuals that have both and have lost total communication. But Wernicke's is understanding speech. So you can say, okay, I want an apple, and someone says back to you, there's no apples in the refrigerator. You can hear that, but it doesn't make any sense to you. All right? So it's processing. It's the association component of processing speech. It's like hearing a foreign language. So nothing wrong with the sensory area of sound. You hear the sound, but it's a nonsense sound. All right? And so you can't, unless there's reading, you can see the words. So with the same damage, you can see the words. But again, it's like reading a foreign language. So it's not just speech, it's any internal it's any communication. So if you're deaf and you're signing, you can't understand the signs. If you're reading, you can't understand the words, all right? So pictures, you'd have to communicate with pictures rather than symbols. So it's the understanding of symbols. However those symbols are presented, translating those symbols becomes difficult or impossible with damage to Wernicke's area. And I believe that's it. Yeah. Basal nuclei we'll talk about when we talk about the thalamus. We're not identifying them in labs, but I want to talk about their general concept. We'll pick that up when we get back to the spring break. So I will see most of you in lab. If I do not, the Dr. Lancaster's group, remember Monday is a holiday after spring break. So at Sac-State, so we have our next class is going to be 2 weeks from today. So do a little studying this week. 16 is what we covered in lecture this morning. And then I'm going to go ahead and go over with you the thalamus and hypothalamus as well as brainstem. Although we haven't lectured on that yet. We'll do that on when we get back. So I won't see you guys for 2 weeks. Because next week is spring break, and then the following Monday is Cesar Chavez Day, which, so we're missing one of our days, and then we have class on Wednesday. We still end up having 5 days for this unit. Typically, every 5 days we have a test. So we were scheduled to miss a day. There's just no way I could have covered this in 4 days, and it's not material I want to tell you to study on your own. But what that means is we have our last lecture lab day on a Monday and then no weekend in between and our exam on Wednesday. Not the week we come back, but the following week. So you need to plan ahead. Take spring break, you guys need it. But that Cesar Chavez Day, if you can, I would turn that into a study day. All right, so look ahead and see your schedule because we're not going to have a weekend between learning the eye and the ear and endocrine system and then we have our test 2 days later. So plan ahead for that.
Wait, but our test is on Wednesday.
Our test is on a Wednesday. So we come back on a Wednesday. That day we'll do brainstem and cranial nerve. Then we have a weekend. And then that Monday we'll do eye, ear, and endocrine. And then that 2 days later we have our test. Okay, so you need to so that's why I'm kind of moving ahead a little bit and going over brainstem with you today, even though we won't have covered it in lecture. So when we come back, the day we come back, we do brainstem, cranial nerves, and a brain dissection in lab. So all of that is why I'm going to go ahead and do brainstem with you today, even though I haven't lectured on it. So it gives us a little bit more time in lab. We can kind of move maybe part of the special senses up a little bit so that last day you don't feel so crammed without some time for that to settle in. So look ahead at your schedule. All right, if you can study during spring break, that would be great. I was a student who loaded up my suitcases and flew home with all my books and never opened them. But I did feel guilty at least. But at least that Monday, all right, plan on that being a study day. Our next quiz, because we have the brain dissection, I don't have a quiz scheduled for the Wednesday that we get back because our day is going to be full. So our next quiz is going to be the last day before the exam, and it's going to cover at least one ventricle structure question from Monday. It'll cover brain, brainstem, and cranial nerves, and it's designated that way on your schedule. Okay, so again, kind of keep up with that material. So we're on page 118, 119, and we'll do page 120 and 121 as well. There's not much to page 119, okay? There really isn't, so I don't find that too difficult to follow. So the first structure is just looking at a gyrus. Remember I said a gyrus is just the fold of gray matter. The sulci are the two valleys between that fold. We have a couple of gyri that you need to learn the names for, but just as a general term, that's what a gyrus and sulcus is. If you look at this cut surface of the brain, this would be a gyrus, and the groove on each side would be the sulcus. Right? For those specific components, on your diagram, F and G are the pre- and post-central gyri, and H and E are the sulci that we're going to learn. So now when you look at the diagram on the lower left, is showing the brain on an inferior view like this. So you have to flip it over to think about it. So when you're looking at it like this is going to be the right hemisphere, and this is the left, if you think of it the way it's going to fit in the brain. So L is showing the right hemisphere, and M is showing the left hemisphere. Remember the cerebral hemispheres from our embryonic development come from the telencephalon. So if we're matching up lobes, frontal lobe, it says that on your lab manual, so the word telencephalon is right there in parentheses by cerebrum. So you would be matching frontal lobe, precentral gyrus, temporal lobe, lateral ventricle, all of that are components of the telencephalon. Now, if we separate the cerebral hemispheres, all right, in this fashion like that on the sheep brain, when we do our sheep brain dissection, you'll be able to do that, but not completely without cutting it, because the two cerebral hemispheres are going to be joined by the corpus callosum. But when we do separate them, that space is identified as the longitudinal fissure. That's letter I on your diagram. So I can mark that either by indicating right here, identify this space, okay, between the two would be the longitudinal fissure, or I might slip a piece of paper into this gap, like that, and say identify that space, and that would be longitudinal fissure, okay? Then the sulci are visible on the lateral view. So the lateral sulcus, you don't have to include the word cerebral, but I would mark it wrong if you did. Lateral sulcus is fine. And that is this one right here. So that's going to separate temporal lobe from frontal and part of parietal. And that is letter E. Now, the diagram on the right with letter E shows the cerebellum, so it makes it easier to figure out that that's the posterior aspect and this is the frontal aspect. The diagram on the left doesn't show the cerebellum. So in figuring out which is frontal lobe and which is occipital lobe, you need to look at that cerebral, that lateral sulcus, and that will help you orient yourself to which is front and which is posterior, anterior and posterior in the brain. The central sulcus is passing between, separates frontal lobe from parietal lobe. And on some of the brains, that has been indicated by highlighting with pen and pencil. Okay? On other brains, not so much. So on this one, it really isn't showing. So I would not take this brain and point to this sulcus and say or point to this gyrus and say identify this gyrus. Because then you're left trying to decide if this is the central sulcus, or if that's the central sulcus, or that's, so which side of the sulcus is that gyrus? So on something like this, I would point to the sulcus as well. And say, identify this sulcus, and then the other question would be to identify the gyrus, and that way you're oriented. Because the only sulcus that's there is the central sulcus. And if I asked the gyrus in front of it, it would be the precentral gyrus, and the gyrus behind it would be the postcentral gyrus. Okay? If it's been marked like this one has, okay, where the sulcus is plainly marked, then I might just say identify the gyrus. Or on the chart over here, where the two gyri are shown in different colors. So the yellow gyrus would be the precentral gyrus, and the bluish gray one would be the postcentral. So I could mark those without asking for the sulcus. Okay? But if the sulcus isn't clear, I will not ask for the gyrus without also tagging the sulcus. So on your diagram, the central sulcus is H, precentral gyrus is F, and postcentral gyrus is G. Precentral gyrus is motor or sensory? Motor. Good, because it's in the frontal lobe. And that's what's formed of our upper motor neurons. Postcentral gyrus is sensory, because it's in our parietal lobe, and that's where we process pain, touch, temperature, sensations. All right, if you would add another sulcus, in lecture I talked about the parietal occipital sulcus, and so if you want to draw it in on this lower diagram right here, you would highlight this a line right across here, and that separates parietal lobe from occipital lobe. So kind of the last third on the hard plastic models is pretty clear. It's right here. So this right here would be the occipital lobe, and that would be the parietal occipital sulcus. So it's spelled parietal with an O, P-A-R-I-E-T-O, occipital. O-C-C-I-P-I-T-A-L. All right, so right there. And that gives you on the medial surface the best separation between occipital lobe and parietal lobe. Okay? On the lateral surface, it's unclear. It's only like an inch or so across the side. So you can see it best on the medial surface. Okay. All right, the corpus callosum. Anybody remember what type of white fibers the corpus callosum is? Association, commissural, or projection? Commissural, good. And this is corpus callosum right here. So these are fibers that are traveling between the hemispheres. It looks like a structure that goes this direction, but they're actually fibers that are going back and forth like this. So if I were to take back of this sheet, so the corpus callosum is going to be, oh no, this is somebody's quiz from the next lab. So those would be white fibers connecting the two halves, and they form the corpus callosum like that. And if you see it on the side view, the way we see it in the brain, it looks like that. Okay? But the fibers are actually going left to right, right to left. So corpus callosum is letter N. N is in nevermore. If we look, I'm going to come back to olfactory bulb. So if you look at the lobes now, and they seem to be pretty basic, but you'll be surprised somebody always misses it because they're not used to the surface I look at. So we'll look at all the surface of the lobes. So the frontal lobe is not just this front part here, back to the central sulcus. It's also what you see here. So on the inferior aspect is frontal lobe. And then if we look at the medial aspect, here's cerebellum, so that tells you it's posterior. Frontal lobe is also here. Okay? So basically all of this that I'm grasping is going to be frontal lobe. You should recognize it from any of those views. And that would be letter V on the diagram in the upper left. Okay, temporal lobe is inferior to the lateral sulcus. Its most posterior definition, there's not a distinct sulcus that separates it, but all of this, and then on the inferior view, this right here, the bump, this inferior bump here is temporal lobe as well. So essentially what I'm holding here would be temporal lobe. Hippocampus is going to be inside that. And while you don't have to identify hippocampus, we'll see it in the sheet ring. It's really cool. So that would be letter D, as in determined, in the upper left portion. Parietal lobe is going to be posterior to the central sulcus, back to about here. All right. So this portion will be parietal lobe. And when we look at the medial surface, we don't see the temporal lobe on the medial surface. It's too lateral. But parietal lobe is going to be basically where my fingers are here. So that's parietal lobe. Okay. Frontal lobe here, parietal lobe here, and occipital lobe here. You're not going to see temporal lobe on this medial view. So parietal lobe would be parietal lobe would be A, and then C would be the most posterior aspect. Again, the parietal occipital fissure, and essentially what I'm grasping here. So from the inferior surface, it would include this part back here. This would all be temporal, but back here, essentially that portion would be occipital. Okay. And when I tag it, I'm not going to go, tease you by kind of putting it right somewhere in between the two. I'll make it distinct. So C is occipital lobe. And then the insula is the shaded area, O. So on the hard brains, you can't see the insula because you can't open up the lateral sulcus. But if I open up the lateral sulcus like this, you can look inside, and see that there's cortical tissue there. It's folded into gyrate. And so what you're looking at, if I pull this away and bring this out, is this structure. That's the insula. All right, so on our isolated brainstem models, that's insula right there. And that's probably how I'll tag it, because it's hard to keep that lateral sulcus open to tag it down the base. But that would be the insula. And it's the shaded area marked O. All right, so that's what we covered in lecture today. So if you'll turn to page 120, 121, we'll look at the diencephalon. Remember that diencephalon and telencephalon developed from our early embryonic three vesicles, the most anterior being the prosencephalon. So telencephalon is going to be the lobes of the brain, the insula, the lateral ventricle. Diencephalon is thalamus and hypothalamus, which are covered up by the rapidly growing hemispheres, as well as the pineal gland and pituitary gland. Those are part of the endocrine system that we'll cover the last day before the exam. So you already should be familiar with the thalamus and hypothalamus from the lateral view that we looked at on Monday, because that space is the third ventricle. All right, so the third ventricle is the space between bordered by thalamus and hypothalamus. So if I had this model on a lab exam and I wanted third ventricle and I tagged the hypothalamus, if I wanted third ventricle, I'd have to say identify this space. Otherwise I want the structure itself. Okay. So thalamus is going to be the rounded region. So it's letter C on the upper diagram on page 120. And the circle in the middle of C, letter B, is the intermediate mass. That's not formed by bridging fibers, by commissural fibers, but just two parts of the thalamic nuclei that happen to touch each other. So remember in our ventricular models, when we looked at the third ventricle, on this model, there was a hole in the middle of the third ventricle. That's actually a solid structure. It's a solid structure. The third ventricle is a space. So that hole in the middle of the model is that intermediate mass, where the two thalamic nuclei touch each other. All right. So B is the intermediate mass, C is the thalamus, and that's all you can really see of the diencephalon, thalamus, and hypothalamus in this model. We'll get to the hypothalamus in just a moment. But on this model, there's the thalamus, go posterior, and you can see a much larger region of the thalamus posteriorly. Okay? So be sure and look at that with one of the brainstem models. Now I have a really large brainstem model here. This is the front. So all of this structure right here, looks like an egg. All of that is actually thalamus. Okay? And we'll talk about the function of thalamus in a couple weeks. The hypothalamus is inferior and anterior to thalamus. So hypothalamus is letter A on your diagram. Growing out of the floor of the hypothalamus is going to be the optic nerve and the pituitary gland. So some of these models have a orange or brownish ball kind of hanging down from a wire, and that's the pituitary gland. On our sagittal head model here, you can see the pituitary gland within the cella turcica. So part of the pituitary grows out of the floor of the third ventricle from the hypothalamus. Part of the endocrine system, and we'll come back to that again the last day of lecture. Part of the optic nerve comes back and forms a crisscross. That's cranial nerves. Again, we'll talk about that when we get back. But on this diagram up here, so when you look at the ventral surface of the brain, you will see the two optic nerves coming back from the eyeball. towards the center, and part of the nerve fibers will cross and go to the other side of the brain. Part of them do not. And they do this pattern. So this is optic nerve. We'll talk about it when we get back because it's part of the cranial nerves. Where some of the fibers cross is identified as the optic chiasma. And it's seen as letter F on the upper diagram where it's been cut and you're looking at a sagittal view of that cut surface. Okay? You cannot see it on the lower diagram because it's covered up. You can see the X, but the optic chiasma itself is covered up by the stalk of the pituitary gland. And then O would be the optic tract. Now these are sensory fibers, and I already talked about the thalamus being a sensory relay. So all of our sensory information except for smell, those neurons go to the thalamus and synapse with other neurons that if it's hearing will go to the temporal lobe. If it's smell or pain or touch or temperature, not smell, vision will go to the occipital lobe. Pain, touch, and temperature, parietal lobe. All right? So if you follow on these brainstem models, it's a little harder on the whole brains, but on the brainstem models, if you follow that optic tract back, it'll take you to the posterior thalamus. It'll take you to this large fat part of the thalamus. So optic tract, you can just follow it back and around and end up in the thalamus. It helps you find that. Okay? Again, you can't do it on the solid half brains. You just can't get to that point. Alrighty? So O is optic tract. Mammillary bodies are part of the hypothalamus. Remember I talked about hippocampus and the temporal lobe? And when you lay down short-term memory, there are nerve connections between the hippocampus and the mammillary bodies. Okay? Mammillary bodies are also associated with a part of the brain known as the emotional brain. Interestingly enough, if they're damaged, people show abnormal behavior, a lot of licking. So it's part of, we don't really talk about much of those structures, but that part of the mammillary body. So from the ventral view, you can see them in cross-section. It's not labeled in cross-section, but in the ventral view, they're numbered letter N. And so they're found just between the optic tracts. Somebody thought they looked like small breasts. And so they're called mammillary bodies. All right? And I'll show them to you on this large brainstem model. On some of the smaller brainstem models, they're also easily visible. The other, again, the large brains, they're kind of compressed and that space is tight to see that area. So now let's move into the midbrain. The brainstem is divided into midbrain, pons, and medulla. So again, we see our motor structures are primarily ventral, and sensory structures are primarily dorsal. So from the front, we can see, my way here. Remember I talked about the motor pathways being two neurons, the cell bodies. If I decide I'm going to extend my fingers, cell bodies in the precentral gyrus fire, and that sends an upper motor neuron down through my brainstem, it crosses over and goes down my spinal cord, and synapses with lower motor neuron cell bodies in my anterior gray horn. So all those fibers descending down form a bundle of upper motor neuron fibers coming down to the front of the brain. And that forms a structure forming a V on the ventral surface And this is known as a cerebral peduncle. It's part of the midbrain. And it's a bundle of projection fibers. These are fibers that are projecting from the brain to the spinal cord. And they're just a bundle of upper motor neuron fibers. So let me take this large brain model. Again, this is ventral surface. All right. So all of this is cerebral peduncle. Those are upper motor neurons. And right there are the two mammillary bodies. So those are going to be found in the V created by the cerebral peduncles. So my fingers are on the cerebral peduncles pointing to the mammillary bodies. Okay, again, ventral, cerebral peduncles are motor. Okay, right there. Did you see it? Okay, and then my fingers are pointing to the mammillary bodies. So these are the peduncles. Those two bodies in the middle are the mammillary bodies. Okay. So mammillary bodies, and I'm pinching the cerebral peduncles. Okay, and mammillary bodies are those two bumps in the center. So pinching the cerebral peduncles, those two bumps in the middle are the mammillary bodies. Right here. So pinching the peduncles, and those two bodies right here are the mammillary bodies.
So these are the peduncles there?
These are the peduncles right here. Right.
What did you say we wanted to see?
Those are upper motor neurons. Okay?
Yeah. These are the peduncles.
All right, now, notice that just inferior, so in your diagrams, the cerebral peduncles are G. I'm not going to tag them on a brain model like this. It's just too hard to see down in there, all right? So I'll use a brainstem model like this or one like this. They're a little bit easier to see. Now, so inferior to that, we have this large rounded, I call it a pregnant belly or a beer belly. The reason it's large is because not all of these fibers pass all the way down to the spinal cord. Some of them will stop and synapse with fibers going to the cerebellum. So the presence of these cell bodies creates the large swelling of the pons. It's actually that surface is the basal pons, but it's the ventral surface because it's motor neurons. All right. So we see this area here, ventral, which is basal pons. If you turn it over, the area behind it is the open fourth ventricle. The roof over that would be the cerebellum. Okay? So there is no dorsal structure for the pons. Which reminds me, I skipped the dorsal structures for the midbrain, so let me go back to that one. If you look at the posterior surface of the brainstem, you'll see two swellings. Now, if you're looking at a sagittal view like this, find the cerebral aqueduct that we studied on Monday, and you'll see two little bumps above it. So here's the cerebral aqueduct, and here's one bump and another bump. Okay? If we put the two halves of the brainstem together, we actually have four. Two on the top and two on the bottom. This is a posterior view, or a dorsal view. And so those, here's my other half of the brainstem. Is there a brainstem back there that's two parts put together?
Yeah.
We have a complete brainstem and then two halves that,
So it's going to be
there we go.
superior.
Okay.
Inferior, pocket.
Yes. So now, this is the posterior view, fourth ventricle, superior, inferior colliculi. Okay, and you will see that on the sheep brain as well. So, superior colliculi, inferior colliculi, fourth ventricle. Did you see it, Todd?
I didn't.
Okay. So, superior colliculi, inferior colliculi, fourth ventricle. Fourth ventricle is shared by pons and medulla. Superior and inferior colliculi belong to the midbrain. Superior colliculi, inferior colliculi, the diamond-shaped opening is the fourth ventricle. That's the only posterior structures you need to identify for the brainstem.
I just
Okay.
want to relax.
So again, superior colliculus, this would be thalamus right here, like I drew on the board. Superior colliculus, inferior colliculus, fourth ventricle. So you'd have to turn this over like this. Superior colliculus, inferior colliculus, fourth ventricle. Okay? Superior colliculus, inferior colliculus, fourth ventricle. Okay? Now, I haven't given you much function, but I just need to warn you, on spring break, I'm not sure where you're going, we're headed for San Diego, so it's not supposed to be warm, but let's say it's warm and I'm out there on the beach, haven't seen my husband in a couple of weeks, so we're laying out there, you know, reading our summer novels, and this guy goes walking by. Now, my husband's pretty good shape, okay, for being 60, but let's say that somebody in better shape goes by, and I can't help it, I just start to follow. I can blame it as a reflex on my superior colliculus, because the superior colliculus is hooked up with that optic tract. And when something crosses my field of vision, there's a reflex of movement that my eyeballs and or my head turns to follow. Okay, so if you're out to dinner and the same thing happens, blame it on your superior colliculus. The inferior colliculus is involved in a reflex where you turn your head for sound. So you hear a sound and you just automatically turn to hear better. Okay? So those are sensory reflexes, superior, inferior colliculi, dorsal part of the midbrain. So superior colliculus is shown in the sagittal view as letter D, and inferior colliculus as letter E. All right. Let's skip the cerebellum. We just did the pons. We'll do the medulla, and then we'll go back to cerebellum. So remember these motor fibers? The upper motor neurons formed our cerebral peduncles. They pass through the pons, synapsing with cell bodies that go to the cerebellum that make the swelling of our pons. But then they keep passing inferior, right? So here's the bottom of the pons, and those motor neurons keep descending down. There's fewer of them because some of them stopped in the pons. And so these create, in the medulla, a longitudinal swelling known as the medullary pyramid. And that would be letter I in your diagram. Now, I will lecture on this on Wednesday when we get back, all right? Because the afternoon class, like Hester, I'm not sure how much of that he'll go over. But I wanted you to be familiar at least with the structure, so we don't have a ton of material to go over new. So just below the pons, on the ventral surface, because these are still motor neurons, we have the medullary pyramids. On our big brainstem model, it's these two struck columns right here. Okay? Discontinued by the same fibers that form the cerebral peduncles, pass through the pons, and now form the medullary pyramids just below the pons. Letter I. Now, Dr. Carter has also put in the lab manual the olive, which is letter J. We're not going to talk about the function, but she put it in there to help you find the hypoglossal nerve, the motor nerve for the tongue muscles. So just lateral to the pyramid is a small oval swelling that looks like a large olive, or small olive, depending on the size of your model. That's letter J, and the hypoglossal nerve we'll talk about and when we get back is the nerve fibers between those two. All right. So those 2 pages, okay? And then we'll do the lecture concepts when we get back. Oh, cerebellum, thank you. I forget to do that. So here's the cerebellum. We'll talk about its function when we get back. It's primarily for balance. These would be the cerebellar hemispheres, the lateral large aspects. Cerebellar hemisphere, it doesn't show both, but L would be the left cerebellar hemisphere. There are 2. They fit together like this. Okay? And then between the 2, but that way, they fit together like that. In between the 2, more shallow, is the vermis. So the vermis is letter M. Right Right, so it's like we have a so here would be a cerebellar hemisphere, here would be another cerebellar hemisphere, and the vermis is this middle region. Right, it's been cut in cross section. So that's letter M. Now when we cut through the vermis, you can see a pattern of white matter that reminded somebody of the branches of a tree or maybe the root system. And so it's called the arborvitae, which means living tree. All right, so that's the white matter pattern shown as letter K. Now I'm done talking. Unless I'm answering questions. But those are the 2 pages that we're covering today. And then I will lecture on them. We'll go over them again when we get back.